We have our next speaker, Eivind Almaas. He's a professor at the Department of Biotechnology and Food Science. He's a group, and several of his members are here, so he's involved in several of the digital projects, actually. His group is working on systems biology and also synthetic biology. And today he will talk about a method to study global scale models around. So please. Yeah.
All right, it's a great pleasure to be here and have the chance to talk about genome-scale metabolic modeling. So I was very happy when I received the invitation from the organizers to have the chance to talk about this project that we have recently completed. And this is a method that we call the RAMP that focuses on how to combine uncertainty in data with genome-scale metabolic models. But before I get into the details of it, I'd like to introduce you to the work happening in my group and then some basics of genome-scale metabolic modeling. So in my group we have three major focus areas, two of them taking most of the attention and time right now. Network analysis, where we study different types of large-scale networks. They don't have to be only biological in nature. Some of them are also technological, social, relationships networks. But the majority are different types of large-scale biological networks. And going over to metabolic modeling, that is also, of course, a network. And when we do genome-scale metabolic modeling, that's one technique that is very powerful. I hope you'll agree with me when the lecture is over, in studying properties and phenotypes of cells. In addition, we also think of it as a network and use a variety of network approaches to study these systems. Finally, we have done some work in stochastic analysis of genetic circuits. So that's also some sort of a network, but much smaller scale. And last but not least, Ibm Competition. It's a student competition in synthetic biology where teams of students all over the world get together and they decide on their own different projects, and they meet up in November, typically in Boston, and have a nice jamboree competition in a friendly way. And NTNU has had a team for now 7 years. And this year's team is focused on studying phages and how we can evolve phages to target specific bacteria as an alternative for antibiotics. Flux balance analysis. Now I'm almost a little uncertain how I should present this, since Bernard Paulson is in the room. And several of the graphics that you'll see in my presentation are from his past papers. But I decided if he's not going to do a quick intro in his Volterra lecture, I need to do it for my talk. And in any case, if it comes in the Volterra lecture, it will be too late since my talk is now. So here we go. What is flux balance analysis? That is the most common approach for modeling genome scale metabolism. And it's based on a few very simple principles. So simple, in fact, that you might look at it and say, how is it possible to get any biology out of it? It is so simple. You know, somebody out of high school could do this. I'm exaggerating, but not really. So you say you have a space of metabolic fluxes, and then you impose parts of reality on these fluxes by saying, these fluxes are really measurements of activity in reactions. a reaction doesn't happen by itself. It produces something that is used in another reaction, which produces something that is used in another reaction, etc., etc. They're all interconnected. That connectivity is captured in what we call the stoichiometric matrix, which is tallying up how different metabolites are connected in different reactions. And this is a constraint on the system, and when we have variables, we connect them with constraints. We call that equations. And that reduces the freedom of the system from being able to have any point to be limited to only those points that satisfy these equations. Now, in these systems, we have many degrees of freedom. So what we do, we are posing particular questions, saying, what type of solutions will give us something that gives biological meaning? For instance, maximal growth, maximal ATP production, minimal something. And that is picking out certain regions or points in this solution space. So that's what this picture captures. And when we put it in a list of bullet points, it is first tally up all of the metabolic reactions that are present. Then you apply a principle of mass conservation. That doesn't sound too bad. Then steady state. Well, that might be a little bit shaky. And finally, we say the system is optimizing. That may also be questionable. In many situations, we can argue very well that it is optimal. Other situations, it may be a bit difficult to say exactly what is optimal. But if we just go with these basic tenets, we get many great results. So first, a little weighting slide to show you some of the details in the system. I'm pretty sure most of you have had some contact with metabolic networks in some coursework. And you can represent it in this following way. You have fluxes coming into the system, carrying metabolites that are used in combinations to produce other types of metabolites. And then you just represent it in this stoichiometric matrix. So reaction V1, you read down this column, and A and B are used, and D is produced. It's no harder than that. And when you see how easy it is, you know immediately this is something a computer can do. And there are a lot of good tools for automatic model building as long as you have a good reconstruction or a good annotated genome. A starting point, a blueprint of what are the metabolic reactions present. And even some of the methods for generating models, they're able to do hole filling and guessing based upon templates from other organisms. So we don't have to have the perfect annotated genome, but of course, the better it is, the better your result will be. Now, what can this modeling paradigm do for us? And this is a paper that really made me pay attention from Paulson's group in 2001, looking at growth of E. coli on acetate and in presence of oxygen. And this is the theoretical surface, the space of possibilities. And you see these blue dots, that's where reality turned out to be. And if you look in 2-dimension, acetate uptake versus oxygen uptake, and we see this shape where we predict that growing states should be possible. And this red line corresponds to the best way of combining the carbon source and the oxygen. That's where I get most bang for the buck. And as you see, nature has found that place, that sweet spot. So that's an indication that yes, there is optimization going on, and that this very simple framework is able to come with very high quality predictions of what is happening. Now, in addition to predicting growth phenotypes, you can use it for a large variety of studies, gene knockouts, how they perform. Different types of analyses have different levels of accuracy, depending upon, of course, the model reconstruction. Now, I'm claiming that this approach is very accessible. There are tools for doing all of these analyses so that you don't have to be an expert in programming or all of the different steps involved. If you have a basic understanding of what is going on in metabolism, you can start playing with these models within, for instance, the Cobra toolbox in MATLAB or in the Python package CobraPy. Take a model and start massaging it to see what type of phenotype predictions you get out from it. So, in addition to these very developed projects, there is a variety of standalone packages that can do some specialized functions and more general functions. In addition, this fall, the Digital Life Norway and Norbis researcher schools, we will have a course on genome-scale metabolic modeling. I would like to announce it to you. Unfortunately, the deadline is, the registration is over and we're fully booked. Well, not unfortunately, it's a good thing. For those of you who have registered, I look forward to seeing you there. Now, what is serving as a bridge over to what I'll be talking about as the main focus of this talk is this last statement that sort of snuck in there. Many of the approaches have been developed for combining, for instance, omics data with metabolic modeling. What does that mean? Well, we have a framework that is optimization based. That means it's easy to add different types of constraints to the system. But how we represent the constraints and where the information comes from says something about how we need to deal with them. For instance, this stoichiometric matrix. In most of the reactions, we know there's only one of this molecule combining with one of the other molecule. There's no uncertainty in this one. It is not 1.1, it is 1. Why should we care about uncertainty in the data? Well, if you want to do more advanced modeling, you pull in, for instance, omics data. And you can use those as constraints. And that means you have data into your model that are associated with uncertainty. So, in addition, we also have some of the basic assumptions that is steady state and that all experimental measurements are performed on populations that are identical and performing optimally. Is it possible to relax these constraints and explicitly handle the uncertainty and variation that we know exists in these systems? And that's where robust analysis of metabolic pathways, this RAMP, is a framework that can answer to these challenges. So now, just a little warning. We will get into some uncertain waters. But I promise you, at the end, you will see that this approach can give interesting results. Now, mathematical development. And for those of you who are not too familiar with mathematical formulations, I put in this 1 of 2, so you know when this will end, so do not despair. This is the basic assumptions for flux balance analysis. We say this flux is built up of contributions from many different, the change in the concentration of this metabolite is built up of contributions from many different fluxes, of different reactions. And we set that to 0. This is standard FBA. Now, if we instead say, this is not just one idealized cell, but we have a population of cells, then we can say this change of concentration, this rate of change, comes from a population average of this collection of cells. What does that lead us to? We take the central limit theorem, and then we say, well, for these populations, we're not talking about, even if we do subsampling, we're typically not talking about a handful of cells. We're talking about thousands, millions of cells. So we can reasonably expect that this is normally distributed, approximately, from the central limit theorem. So in order to then clamp down on what is the variation that we can't tolerate away from steady state assumption. We say that this rate of change is allowed to be bigger than some bound by a small probability epsilon. And it's allowed to be smaller than the negative of this bound, so it's outside of this bounded region with a small probability epsilon. That's where we say the system needs to be. So these are bounds on the tolerable variation. If these bounds M go to 0, we're back at flux balance analysis. Now, saying that this is some average over populations or is a representative of a collection, we can say, what is the average flux of this change of this metabolite? Well, it's of the population, and this is something we can rewrite as a stoichiometric matrix times the flux, and we represent that as the estimated value of the stoichiometric matrix. So now we're transferred the variation into the stoichiometric matrix to say that the variation is in the stoichiometric coefficients. This last step is using something called scenario analysis, saying, well, how should we break down this stoichiometric matrix and the possible variation you can have in it? We will break it down into distinct scenarios, and this is an approximation. And in these distinct scenarios, which we will associate a probability with, then we can say we have, for instance, 3 scenarios, 3 possible values for a stoichiometric coefficient. We can have 10 different values. We can think of it as fine-graining the distribution of possible values. Also, using this approach, we can write down what the variation is in the flux. In many situations, when we analyze the system, we do expect that the average will still be 0 in RAM. However, now we can explicitly get a handle on the standard deviation in these fluxes around the expected value of 0. So, introducing scenario analyses in these expressions, we can rewrite it as this R. I'm not going to talk too much about that. If you're interested in details, it's all in this paper that we got out earlier this year. But this is where scenario analyses are hiding. So inside here, there's some P's for probability, and you're assuming there are multiple states. And then some more math happens. Again, look in the paper. I don't think it's directly relevant for this presentation. And then we have the final presentation of ramp as a method. So we maximize an objective. In this case, it's exemplified as growth. Doesn't have to be. And we'll see some other applications in a few slides. We have stoichiometric matrix multiplied by flux, but instead of set equal to 0, we say it must be smaller than this bound, and then we have some conditions on what the stoichiometric matrix can be. Because this is no longer a fixed stoichiometric matrix, but a set of stochastic variables drawn given this condition. And this is the scenarios, and say that the scenarios must be within a certain distance where this u variable is bounded. And that type of formulation we get is something called a second-order code program. Now, to contrast with standard flux balance analysis, we see some of the parts are the same, the bounds are the same, but this part, the simple part of steady state, has been exchanged with this part where we say we have in fact, a stochastic matrix that must be bounded. And these are the conditions for the bounds. Wow. To make it a bit more accessible, I'm going to display this example. So assume that we have one metabolite, it's a very simple and cartoonish to the extreme metabolic network. One metabolite and 2 reactions. How could this system be set up? Well, for flux balance analysis, this is the definition of the system. The fluxes, we say, are bounded between +1 and -1. And the solution space, the null space, is this dotted line. It goes from -1 to +1. So that is our solution space. Now when we introduce ramp, we need to come with some definitions and make some assumptions. For instance, we say the stoichiometric matrix isn't just 1 and -1, but we allow for some variation. In this case, let's say the first coefficient could be 0.9 with a probability of 25%. It is 1, what it was originally, with 50% chance, and it is 1.1 with a chance of, again, 25%. And I want you to notice that the variation is on the same scale or level in the second row. Then it's from 1 to 1.2 and 0.8. Setting up this and churning the machinery, we get that this gray region is the solution of the problem. The solution must be within this gray region. And as you can see, it doesn't fully encompass the flux balance solution. There is an overlap, but it isn't including it. So this means that we can't just say that this is an extension. We get a bigger solution space than the flux balance analysis, which includes the flux balance analysis. It is indeed something different. But many of the solutions you find in FBA, you will also find among the ramp solutions. This family of lines, these are just to indicate that the constraints that you see, these constraints are setting up this infinite set of lines. One of the constraints for the upper half and one of the constraints for the lower half. And for different u values, you get different of these lines. And the solution is on the inside of that area. Now this is all well and nice, but how does ramp help us understand metabolism? Since this is cleanly formulated mathematically, we can crank the mathematical width at the problem, and we've developed some theorems. I'm not going to pose them mathematically correct, but please allow me to paraphrase them. And again, go to the paper if you want to see the exact formulation of these theorems, and we also prove these theorems. Now, the ramp optimal fluxes are continuous with changes in parameters. So what can I say? Well, with standard FBA, you may have the concern that when you change different coefficients in the stoichiometric matrix, you may not have a continuous change in the solution space. For ramp, however, we can prove that the changes are continuous. And this is important. A few years ago, there was a question raised by a group at MIT claiming that the whole flux or constraint-based community had made mistakes. We had not solved our models appropriately. In fact, none of the models could be solved. There were just numerical errors as a reason that we thought we were solving them. And this is theoretically possible, but as it turned out, that was not the reason why they weren't able to solve the models. They had a coding error. But for ramp, it isn't theoretically possible to have that problem. Theorem 2. Optimal fluxes of ramp become FTPA solutions in a limit of steady state. This M-bound goes to 0. The ramp solutions become FBA solutions. So you can continuously map into them. And 3. Allowed probabilistic variation in ramp is possible to use that to find what is consistent with an FBA optimal solution. That means you solve FBA problem, and then you can ask the question, I have a solution to the optimization problem. What type of variation in the stoichiometric matrix will still give this as a solution. And RAMP can tell you how much of a variation this stoichiometric matrix can tolerate before the FBA solution no longer is a solution for this matrix. So it's possible to, from this, explore what type of variation is tolerable in a population for as long as this FBA solution is valid. Now, I'll go to some applications of RAMP, because what I've presented so far is purely mathematical considerations. Now, to convert the mathematical framework into a computational model, we have to make choices on exactly how we're going to make different uncertainties incorporated in the system. I'll demonstrate RAMP by calculating fluxes or comparing with experimental fluxes. I look at variation in tolerable variation in growth coefficients in standard E. coli models, and then looking at G knockouts under variation in biomass coefficients. So, in a series of papers, the Sowers group in 2005-2006 measured metabolic fluxes in E. coli for many different conditions. So this is a snapshot from one of the part figures in their paper, and you can see the relative magnitude of fluxes through different parts of metabolism. How do you take numbers like this and compare with what you get from a genome-scale metabolic model? Now, this is a small network, and in the model we use, IJO 1366, we have 1,800 metabolites about, and almost 2,600 reactions. So it's a lot of reactions, a lot of metabolites, and we have few measurement points. So what we're interested in looking at is how close can these experimental fluxes get to predictions from the model. If all is possible to reach all of these experimental fluxes, the distance is 0. It means that the experimental flux point is inside the solution space. However, what we often see is that these experimental fluxes, they're not inside the solution space, they're outside, because these are different networks from also what we're using in the genome-scale model. For these smaller networks, they've had to make simplifications and guesses for how things should be, so that makes up for differences. Now this is a quadratic objective function, because it's a distance minimization, and this is just to show that this RAMP method, it works really for any type of objective function. If you can solve it, that's a question of how good is your numerical solver. And for some of the problems and challenges we threw at RAMP, we discovered that the solvers aren't strong enough numerically. But theoretically, you can formulate this for any type of situation. Now this is what we get when we compare the experimental fluxes, which are black dots and with error bars, to FBA, which are the blue stars, and RAMP, the red boxes. So what you'll see is almost everywhere for these 28 fluxes that are measured in 4 conditions. In A, it is an aerobic batch in glucose for E. coli. B is anaerobic. C is a chemostat on low dilution rate of 0.1 per hour. And in D, it is of the dilution rate of 0.4 per hour. And we see that RAMP is able to track the experimental points much more closely. And in fact, when we quantify it, the mean square error is quite small, except a little bit bigger in this case. And when we do the relative mean square error, comparing it to the best FBA prediction, we see that this is pretty darn good. So, how can we tweak biomass coefficients in a genome-scale model? And the way we do it is you take a coefficient and you squeeze it up and down until you no longer get the same knockout predictions as you had from FBA. So we say we use FBA as the gold standard, and then we push on a system until we get something that's different. Of course, as soon as you tweak the biomass coefficient, you'll get different fluxes. That's why we are interested in looking at predictions for gene knockouts. So this is a big model, and with 1,300 genes, and for every value of the coefficient that we tweaked, we needed to do 1,300 gene knockouts. So that's quite a lot of computations. I'm mentioning this to let you know that yes, RAMP is not quite as fast as FBA, but it is not very slow either. So you can do all of these calculations. So for each of these points, it is 1,300 gene knockouts, hundreds and maybe thousands of times. So we have this line of coefficients, 18 coefficients, and they really should be at 10 to the 26 or bigger. We couldn't get numerical accuracy when we pushed it past 10 to 26. So this just means that for these 18 coefficients, we really have unlimited tweak possibility in the E. coli models. What does that mean? Who are they? Here's a listing of who they are. And when you look at them, for most of them, it is obvious what is going on. These are molecules that are imported from the from outside of the model, directly coupled to the biomass function, and then they don't have any other integration in the model. Which means that when you tweak the biomass function, it is at the coefficient, it is at no cost for the system, and it will just increase the import flux or decrease the import flux. And it's a passive process, so it's no big deal. However, for 4 of them, and right now I can't remember exactly which ones, maybe these 2 and valine and, well, for 4 of them, when I was looking at exactly the import reactions and the reactions that these these molecules were involved in, I wasn't able to trace the simple reason. Because they're involved in many different reactions. And to me that was quite surprising. But in the end I had to take a step back and say, well, clearly it's the same mathematical reason. We get the same result. But exactly how this happens, I'm not able to figure out by looking at the biochemistry. It was getting to be too involved, too many reactions. 54 of the coefficients have limited tolerance. However, that's the tolerance that spans 4 orders of magnitude. So that means that you can tweak these biomass coefficients and still have the same predictions for gene knockout phenotypes as FBA within, for some of them, 4 orders of magnitude. Now the red dots is the multiplier, the scale of the multiplier. The black dots is the actual unperturbed biomass coefficient. In addition, we can also predict gene knockout phenotypes. So then we're testing it against experimental data for E. coli. And here are the computational predictions. FBA is the first line, and then we implemented 4 different ways of doing uncertainty in the biomass coefficients. RAMP 1, 2, and then this 3 ABC, that's just 3 different levels of uncertainty. So in short, this RAMP 1 is using the first significant digit in the biomass coefficient, and that is the level of uncertainty. For 2 and 3, we're doing much more drastic variation in uncertainty. And RAMP 4, then we're saying, what if we are saying it is not important to be close to the steady state? We're just relaxing, allowing to break this M-bound, to go far away from the M-bound. And we see that RAMP 1 and RAMP 4 have almost no, well, they have no effect compared to the FBA prediction. While when we allow dramatic variation in the coefficients themselves, then we see different predictions. So ramp is in fact quite insensitive to breaking of the steady state. That's something I hadn't expected beforehand. So to quickly summarize, I presented a constraint-based approach that explicitly handles uncertainty in the data. It provides a framework for relaxing the assumption of a steady state and allowing for heterogeneity, and able to connect FBA solutions to heterogeneity in populations. What is the level of heterogeneity that a population can tolerate and still have FBA solution as its optimal solution? And we have applied RAMP on actual genome-scale metabolic models, not the smallest we could find, but pretty big ones for single organisms. And I would say it fares pretty well. Of course, when you perturb the biomass coefficients dramatically, you will get different results. But what we see is that there's a large level of robustness in the system. So I'd like to give a big thanks to people in my group who have contributed to the different aspects that we're doing, genome scale modeling and network analysis. Several of them are in the room today. And special big thanks to Al, who's at Rose-Hulman in Indiana, and who's been really the core of this RAMP method. Thank you very much for your attention.
While we're changing, we have time for one question.
It isn't entirely clear to me to what extent are signaling events included in your approach?
In these models, signaling events are not included. So for the typical approaches, we don't include any regulation. It is just the metabolic network.
Okay.
Yeah. There exists some extensions that will include and incorporate. And since it's, as I said, an optimization-based framework, it is possible to include it in a variety of ways.
Okay, so let's take the speed. You have
Yeah, I have also a question. Just to make a story short. So, there are a methodology called metabolic flux analysis, which is localization of the flux balance analysis. And you're using the flux balance analysis as a benchmark to compare your development. But metabolic flux analysis takes into account the variability of measured fluxes on the population level. So now the question arises, how can you first segregate the source of the error of the measurements, either analytical or population level? So in this, now you assume that it's completely comes from the population level. But then the question arises, what is the difference in the RAM methodology versus MFA, metabolic flux analysis? What kind of advantages do you bring there?
So, as I understand it, metabolic flux analysis, it is necessary with different coefficients. Kinetic coefficients is differential equation based. And that is a totally different approach with limitations on what is available of known coefficients. And to my knowledge, it has only really been applied for some pathways that have been very well studied, not for genome scale networks. So one of the nice things of many nice aspects of flux balance analysis, which you just use that as a common name for all the methods, is that you need only few inputs to do interesting analyses. You just need a stoichiometric matrix and some other assumptions of steady state, mass balance, optimization. And that means that this is a type of input that you can quite handily get to nowadays from annotated sequence genomes and then comparisons with existing genomes and what you know of biochemistry. don't need to have these kinetic coefficients.
Okay, let's thank the speaker again.